Thank you so much. Thank you all for your attendance throughout the conference and for my talk. As Matt just said, my name is Lani, and I will be here for you in my research role. So as I look at the impact of prenatal exposure to THC on early brain anatomy and behavior in mice, the work that constitutes the beginning of my PhD. So as cannabis is legalized in increasing countries around the world and states in the US, we see a changing perception both of cannabis itself and of the psychoactive component delta-9 tetrahydrocannabinol, or THC, which I'll be focusing on in my talk. And this changing perception of the safety of THC is not only in the general population, but also in pregnant women. If we look at rates of positive self-report of cannabis use during pregnancy, we see that between 2009 and 2016, they stayed fairly stable. But if we look at positive toxicology, even between this limited time frame, there is an increasing trend. And at this point, that's 6 years ago, as this trend continues. So one of the reasons it's so difficult to assess accurate rates of cannabis use during pregnancy is that it is still heavily stigmatized. So a recent review estimates use from 3 to 35% of pregnancies. This can also differ based on the way that they assess cannabis use during pregnancy and the demographics that are being asked. One more reliable method you can use is a meconium analysis, where they look at the fetus's first poop, essentially, to see if cannabinoids are present. And one study found 13% of pregnancies did have cannabinoids present. However, this is a polydrug sample, so it's potentially higher than other samples. One reason that women might be using cannabis during pregnancy is a perceived effectiveness at reducing the nausea and vomiting associated with morning sickness. So that usually occurs during the first trimester of pregnancy. So that's the time point that I will focus on for my research. As it would be unethical to expose pregnant human women to cannabis, we use a mouse model, and there's important considerations when looking at pregnancy in a mouse model. It's 19 days of gestation and roughly equivalent to the first 2 trimesters of pregnancy. So what's equivalent to the 3rd trimester actually happens postnatally. The endocannabinoid system plays an important role during this development, though, with 2 major endocannabinoids, 2-AG and AEA, being very tightly regulated early in pregnancy. We see low levels of 2-AG and strongly decreasing levels of AEA. So this is the system that we'll be interfering with as we introduce THC to the pregnant dam. So my research question is, what impact does prenatal exposure to THC have on the trajectories of brain development in utero and during the first 2 weeks of life? To study this, I primarily use magnetic resonance imaging. So we can not only extract embryos at gestational day 17 to image them ex vivo in the harvested embryos, but also use it as a non-invasive technique of imaging pups longitudinally throughout the first couple of weeks of development. And I'll talk more about the specifics of this later on. My hypotheses were that early on in utero, we did see increased volume in the regions that develop into the hippocampus and regions that develop into the cortex. And this would reflect underlying changes in neural and glial architecture. Based on previous literature from humans and non-human animal studies, I also expected to see followed by a delay in the growth in the regions that develop into the hippocampus, the striatum, and the prefrontal cortex. One very robust finding also from the rodent literature is anxiety-like behaviors as assessed with ultrasonic vocalization tests, which measure calls that pups make for their dams when they're separated, where we see an increased number of longer calls associated with an anxiety-like phenotype. So the timeline for my experiment comes in 2 parts. I expose the pups through the dam to 5 milligrams per kilogram of THC injected subcutaneously, and we can talk about that route of administration later. Daily injections from gestational day 3 to 10. And then I extracted the embryos at gestational day 17 and imaged them ex vivo. For the pups, I had the same exposure route, but then the pups were born and imaged on postnatal day 3, 5, 7, and 10. I conducted the behavior tests on postnatal day 12, and then the perfusions on postnatal day 13. So moving into the embryo data, you'll see my sample size in the top left, and you'll notice that I not only have the saline and THC group, but also a null group where the dam was restrained but not injected as another form of a control. And then these embryos were actually imaged in the Mouse Imaging Center at the Sick Kids Hospital in Toronto. So I'd like to thank our collaborators, Jason Lurch, Brian Nyman, and Shoshana Spring for making that possible. And then I modeled the volumetric differences in the brains with a voxel-wise whole brain analysis, where we used a linear mixed effects model at each voxel with 2 fixed effects of interest, treatment and sex, and 2 random effects, litter size and coil, because the embryos were imaged on 11 different coils. So here's the heat maps of my volumetric results, which I'll take you through as you'll see them a couple of times. So on the left-hand side, we have coronal sections from the very posterior areas in the cerebellum through to the olfactory bulbs. In the center, we have sagittal sections from the left to the right. And then on the right-hand side, we have axial sections from the ventral areas of the brain to the dorsal area of the brain. So here we're looking at the main effect of treatment. And that means that any voxels that you see in a warm color indicate relative larger volume in the THC exposed pups compared to the saline controls. And conversely, the cool colors indicate smaller volume in those areas. And because we do a linear mixed effects model at each voxel, we have to compare control for the effects of the multiple comparisons. And we do this with false discovery rate. So these are thresholded from 5% to 1%, which is a fairly rigorous threshold. And so we see decreased lower volume in areas like the cerebellum, but a higher volume in areas that develop into the hippocampus and prefrontal cortices. So then we can choose peak voxels from these regions to plot and actually look at the underlying data, which can show us the increase volume in the THC exposed pups. So on the neonatal side, we had to do quite a bit of work to develop the method of the in vivo imaging here in-house. One of the main challenges that we have both in humans, I believe, but then especially emphasized in the mice pups is a lack of contrast between gray and white matter. So in order to counteract that, we enhance the contrast with manganese chloride, a paramagnetic ion. And so previous research suggests that this can really increase the contrast in areas such as the olfactory bulb, hippocampus, and cerebellum. And it's injected in the dam, and then the pups receive it through the milk. We also had to balance the impact of the motion. So they're anesthetized with isoflurane, and we carve these little beds for them out of foam, and we tape them in so that they can remain still during the scan. And previous work by Jason Lurch's group, by Lily Hugh, indicates that the impact of the scanning itself of multiple scan points doesn't impact the body weight trajectories of early pups or the onset of puberty, though it does impact the weight at puberty. So I have non-scan controls in order to further probe the effects of the scanning itself. So that's the reason why my total sample is actually larger than my scan sample. I cull the litters to 6 pups on postnatal day 2, and then include up to 2 males and 2 females per litter for the actual scanning. So the first thing we saw was increased weights in the THC pups compared to the saline controls. And this is interesting because it's a very mixed effect that you see in the literature, both in humans and in non-human animals. One possible explanation is if it's not the THC itself, it could be changes in maternal behavior. So that's something that we're probing further in our data. Looking at the ultrasonic vocalizations, if you were at Lisa's talk this morning, you may recognize these plots. But essentially, we have the THC calls on the top and the saline calls on the bottom. Each point represents a call made by a pup, and the bars represent the deciles, with this bold bar representing the median. So what becomes apparent right away is that the deciles are shifted to the right for the THC pups, indicating that they are making more longer calls. So then using a shift function, we can assess this for statistical significance with each of these points representing the difference between the deciles. The fact that these are positive indicates that the THC calls are longer than the saline calls, and where the confidence intervals do not cross 0 indicates statistical significance. So this is in line with previous results in the literature. It's quite a robust finding across studies. And then moving back into the imaging data, here we interpret the heat maps a little bit differently because we're looking at an interaction between the linear effect of time and treatment. And so, of course, these are neonatal pups. All of their brains are increasing in size over the first 2 weeks of life. But the fact that we see a lot of the voxels in blue indicates that relative to the saline controls, the THC pups are not showing the same high rates of increase. So we can see this more clearly also if we look at peak voxels in the areas that develop into the hippocampus, the thalamus, and striatal areas that we see. Initially, the pups are pretty similar, but they start to differentiate with the saline pups increasing more so than the THC pups. So with that, I'd like to take us through a brief summary. We're looking at the impact of prenatal exposure to THC specifically. In the embryos, we see reduced volume in the cerebellum, but higher volumes in areas that develop into the hippocampus and prefrontal cortex. In the neonates, we see increased weights in the THC pups, an increased number of longer vocalizations indicative of an anxiety-like phenotype, and decreased rate of change as they increase over the first 2 weeks of their lifespan. So next steps for me include looking at the neural and glial architecture underlying these volumetric changes with immunohistochemistry. And then long-term, I will also be expanding this later in the lifespan to look at adolescence, a second hit of THC during that period, and changes in adulthood. With that, I'd like to thank you all for your attention. And if I have time, I'd be happy to take a question.
That was great, Lani. If anyone does have a question, we have time for one, I would say. So if you want to use the raise hand function, that way I can identify you to direct the question towards you. If not, I'm going to ask one actually, because I'm curious. So at the beginning of the talk, Lani, first of all, that was really cool data. It's nice to see, there's been a big gap in this area. So it's nice to see people doing this stuff nowadays, finally. At the beginning, you said you had hypothesized that you would have seen bigger hippocampus. I can't remember the other structure you said you would have hypothesized bigger. Out of curiosity, where was that hypothesis based from? It did seem to match your prediction, so I'm just curious.
Yeah, so one of the things that we were reading about was especially how the endocannabinoid system is involved in the pruning of dendritic spines, and how often at the very prenatal periods you see this overgrowth of like rampant, not higher number of neurons, but more processes associated with them. So that's something that I really hope that we'll be able to investigate more fully when we turn to our post-mortem data there.
Okay, so your prediction is this is more of a neuropil effect than an actual cell number effect?
Yeah, that's what I think based on what I've read.
Okay, very cool.